Scientists wouldn't ever have become fascinated with strawberries if it wasn't for the plant pigment, fisetin. It's a promising anti-aging compound and also a so-called senolytic. It also has anti-inflammatory, antioxidant, and immune-supporting effects. Fisetin is a flavonol, a yellow plant pigment that belongs to the flavonoid group of polyphenols. It gives colour to many different fruits and vegetables. Compared to the now famous plant antioxidants like resveratrol and quercetin, fisetin was unfairly ignored for far too long. It wasn't until recent years that researchers became increasingly interested in its medicinal properties. Science teams are currently exploring its ability to slow the aging process and extend lifespan through its so-called senolytic effects. What's more, fisetin has powerful anti-inflammatory, antioxidant, and immune-supporting properties. Despite promising animal and cell-based findings, research is still in the early stages. Only a single clinical human trial has been conducted with fisetin. Some of the benefits of fisetin: it may protect the brain and improve memory. It might delay aging. It helps to control blood sugar. It may have anti-cancer properties, and it may protect the liver and the heart. Many different fruits and vegetables contain fisetin. Food sources with the highest concentration of fisetin include strawberries, apples, persimmons, lotus root, onions, and grapes. Fisetin increases antioxidant defense. It directly neutralizes free radicals and also increases other powerful antioxidants such as glutathione, superoxide dismutase, and catalase. It can block a pathway called NF-kappa B. This is a switch that tells genes to produce inflammatory compounds. An overactive NF-kappa B response is linked to allergies, autoimmune disease, and cancer. Fisetin blocks inflammatory enzymes that degrade fatty acids. It also reduces the activity of a group of enzymes that cancers require to spread and invade other tissues. mTOR's demands for energy and growth can throw your cells into a frenzy. Metabolic waste builds up, and there's no time to clean it up. An overactive mTOR response is associated with cancer, diabetes, obesity, and neurodegenerative disease. By blocking this pathway, fisetin helps to remove waste and enhance cellular metabolism. The most intriguing part: blocking mTOR increases longevity. In fact, blocking mTOR is the only intervention that increases lifespan in all organisms studied to date. This is because once mTOR is blocked, autophagy is activated, which is a process of recycling damaged cellular components. Much of the interest in fisetin has centered around its potential to help slow aging and extend healthy lifespan. Aging is characterized by the buildup of senescent cells. Cells that stop dividing become damaged and start releasing inflammatory molecules. As we accumulate more and more of these senescent cells, the body starts being affected. These cells start damaging healthy tissues. They contribute to many age-related diseases, from osteoporosis and cancer to heart disease and brain disease. Removing senescent cells calms inflammation, improves physical function, and increases lifespan in animals. Certain plant compounds are able to destroy senescent cells without harming healthy cells. In a 2018 cell-based study of 10 such compounds, fisetin was the most effective one. In old mice, fisetin cleared senescent cells and it increased their lifespan by over 10%. Because of this study, researchers got particularly interested in senolytic or senotherapeutic properties of fisetin. Fisetin also extended the lifespan of yeast by more than 50% and the lifespan of fruit flies by more than 20%. Because of these promising results, a clinical trial is underway to see if Fisetin is effective for reducing inflammation and improving frailty and bone health in the elderly. In multiple animal studies, Fisetin restored blood sugar levels of diabetic rats and mice to those of healthy animals, and improved their ability to control blood sugar levels by increasing insulin levels, increasing enzymes that turn sugar into energy, removing sugar from the blood to store as glucagon in the liver, reducing the liver's ability to make sugar from lactate and amino acids. Fisetin slowed the progression of cataracts and protected the kidneys of diabetic mice by blocking inflammation and oxidative stress. It also protected the liver of diabetic rats from high blood sugar levels by increasing antioxidant levels. Another complication of diabetes is the hardening of the arteries and heart disease. In a cell study, Fisetin prevented high sugar levels from causing inflammation in the blood vessels. Inflammation is linked to colon cancer growth, as well as its spread and resistance to chemotherapy. In a clinical study of 37 colon cancer patients undergoing chemotherapy, Fisetin reduced markers of inflammation, which were interleukin-8 and hsCRP. However, the authors didn't report the effects on the tumor growth and the progression. In rats, Fisetin reduced oxidative stress and the growth of liver cancer cells caused by fungal toxins. In mice, it prevented the growth of lung cancer and boosted low antioxidant levels caused by a toxin in tobacco smoke. It reduced lung tumor growth by 67% in mice and by 92% when combined with a chemotherapeutic drug. It also prevented the growth of new blood vessels supplying nutrients to the cancer. Another study found that Fisetin reduced tumor growth by 66% in mice with melanoma. Certain types of prostate cancer are fueled by androgens such as testosterone and dihydrotestosterone, or DHT. Fisetin slowed the growth of the prostate tumors in mice by blocking the receptors for testosterone and DHT on the cancer cells. Fisetin also protected against kidney damage from chemotherapy in rats by reducing inflammation and boosting antioxidant levels. In cell studies, Fisetin caused programmed cell death, and it prevents the growth and the spread of a variety of cancer cell lines. But this doesn't imply the actual anti-cancer effects in living organisms. Older rats given Fisetin experienced memory and learning improvements. Cell studies revealed that Fisetin activates pathways in the brain that are involved in storing memories. In another study, Fisetin prevented memory loss in mice that were exposed to toxins. Fisetin can easily cross the blood-brain barrier in mice. This is important as there is an ongoing debate about whether compounds like Fisetin can reach high enough levels in the brain to improve its function. Fisetin reduced depression and anxiety in mice by increasing levels of serotonin and noradrenaline, which are neurotransmitters that play a key role in mood. Immune cells in the brain, called microglia, are overactivated in neurodegenerative diseases such as Alzheimer's disease and Huntington's disease. This causes inflammation and damage to healthy brain cells. In cell studies, Fisetin boosted brain antioxidant levels and prevented microglia from releasing inflammatory compounds in response to bacterial toxins. Alzheimer's disease involves the buildup of amyloid plaques and tau proteins in the brain. Fisetin reduced levels of tau proteins in brain cells by activating a process that removes these harmful proteins. In mice with amyloid plaques, Fisetin improved memory, reduced inflammation, and prevented the loss of brain cell function. In mice with Alzheimer's, it reduced amyloid plaque buildup and loss of brain cells. Huntington's disease is a genetic movement disorder in which brain cells get destroyed. In mice with Huntington's, Fisetin was able to improve declining physical function and increase lifespan. In amyotrophic lateral sclerosis, or ALS, brain cells that control muscles die off. Fisetin improved balance and muscle coordination, and it increased survival in mice with ALS. Fisetin protects brain cells and reduces inflammation and damage due to stroke in mice and rats. In another study of rabbits, Fisetin prevented loss of balance, lack of energy, and uncontrolled eye movements caused by stroke. Cell studies show that Fisetin reduces the activity of immune cells in the brain that are responsible for the inflammation and brain damage after a stroke. Aluminium is a toxin to the brain, and it's been linked to Alzheimer's disease. In mice, Fisetin reduced inflammation and oxidative stress in the brain that was caused by aluminium. In cells, Fisetin promotes the survival of brain cells by getting rid of damaged or unneeded proteins. In a mouse model of brain trauma, Fisetin prevented seizures by reducing oxidative stress. Fisetin also reduced seizures and death in mice that were exposed to chemicals and electric shocks. It did so by increasing levels of the neurotransmitter GABA and reducing oxidative damage in the brain. By reducing oxidative stress and inflammation, Fisetin protects the brain and improves cognition and mental health. Fisetin reduces high total and LDL cholesterol and triglycerides in rats that were fed a high-fat diet. In diabetic rats, it doubled HDL levels, or good cholesterol, and cut LDL cholesterol levels in half. A cell study hinted that Fisetin reduces cholesterol by causing more of it to be released into the bile. Cell studies show that Fisetin prevents immune cells called macrophages from oxidizing and ingesting LDL cholesterol. When macrophages ingest oxidized LDL, they create fatty plaques that harden the arteries and cause heart disease. In rats, Fisetin improved poor blood flow that was caused by a high-fat diet. Test tube experiments reveal that Fisetin helps to relax and dilate blood vessels, which helps to improve blood flow and reduce blood pressure. Fisetin protected heart cells from oxidative stress, and it improved heart function in rats with abnormal thickening of the walls of the heart. It also protected heart tissues and mitochondrial function from damage due to a heart attack in rats. According to animal and cell studies, Fisetin may help support heart health by reducing high cholesterol levels, improving circulation, and protecting the heart from oxidative stress. In mice, Fisetin protected the liver from alcohol by helping animals process it quicker. It also reduced oxidative stress, which prevents damage. In mice and rats that were fed high-fat diets, Fisetin reduced the buildup of fat in the liver. It worked by increasing enzymes that break down fats and reducing enzymes that make new fats. Diabetes often causes nerve damage and pain. Fisetin reduced heightened sensitivity to pain in diabetic mice and in mice with nerve injuries. It lowered oxidative stress, and it increased serotonin and GABA activity in spinal nerves, which act to relieve the sensation of pain. Estrogen keeps bones healthy. After menopause, low estrogen levels put women at risk of osteoporosis. The rise in inflammation due to aging also weakens the bones. Fisetin improved bone density and prevented bone loss in mice with low estrogen levels and inflammation. In cells, it worked by reducing the activity of bone-degrading cells known as osteoclasts. Collagen gives structure and elasticity to the skin. In human skin cells, Fisetin prevented the breakdown of collagen from UV sun exposure, a key factor in skin aging. It also reduced inflammation and oxidative stress that was caused by UV rays. Applied to the skin of mice, Fisetin prevented the abnormal growth of skin cells, DNA damage, and inflammation caused by UVB. It also reduces the formation of wrinkles by boosting skin collagen levels. Skin inflammation in eczema is typically treated with steroid creams, which often have harsh side effects. In one study, it reduced skin inflammation, swelling, and redness in mice with eczema. Fisetin has the potential to protect the skin from excess UV exposure, which causes skin damage and aging, and relieve eczema. In one study, Fisetin helped prevent Listeria infection by interfering with bacteria's ability to hide from the immune system. In another cell study, it was active against two fungi that cause infections in people with weak immune systems. It impairs the production of a compound that fungi need to survive, called ergosterol. Fisetin may also help with infections caused by parasites. It blocks the activity of arginase, which is an enzyme that some parasites need to protect themselves. Immunoglobulin E antibodies and T immune cells activate mast cell and basophils, which go on to trigger an allergic response. In cell studies, Fisetin prevented T cells and immunoglobulin E from activating these cells and causing inflammation. Similarly, Fisetin prevented immunoglobulin E antibodies from activating mast cells in mice that were exposed to allergens. In turn, it stopped the release of histamine and other inflammatory compounds. In a mouse model of inflammatory bowel disease, or IBD, Fisetin reduced damage and inflammation in the colon by blocking NF-kappa B activity and restoring antioxidant levels. Even at high doses, scientists found no evidence of side effects or toxicity in animal studies. Clinical studies, of course, are needed to confirm its safety. In the lone clinical trial on cancer patients, stomach discomfort was reported in the Fisetin group. However, this might not actually be a side effect of Fisetin. All the patients were receiving chemotherapy, and the same stomach complaint was also reported in the placebo group. The liver uses the same pathway to process Fisetin as it does for blood thinning agents like warfarin or Coumadin. This can in theory increase the effects of warfarin. Fisetin substantially reduces blood sugar in diabetic animals. The combination with blood sugar lowering drugs may further reduce sugar levels. In a clinical study of colon cancer patients, 100 milligrams per day was effective for reducing inflammation. In an ongoing clinical trial looking at the effects of Fisetin on inflammation, bone health, and frailty in the elderly, Fisetin will be used at a high dose of 20 milligrams per kg body weight for 2 consecutive days. This would be around 1,400 milligrams per day for a 155-pound person. There's at least one simple way to theoretically increase the absorption. Take Fisetin with fats, as it's fat-soluble, similar to other flavonoids like quercetin. Fish oil and other oils would enhance the bioavailability of quercetin, and they might do the same for Fisetin. New formulations combining Fisetin into small fat-like molecules called liposomes may be the only effective solution to increase its bioavailability. These greatly improve its absorption and anti-cancer effects. However, they are not yet commercially available.